where I live.
8.2 says the free energy change of a reaction tells us whether or not the reaction occurs spontaneously. A living system's free energy is energy that can do work under cellular conditions. The change in free energy, represented as delta G, during a biological process is related directly to enthalpy change, represented as delta H, and to the change of entropy, delta S, so that delta G equals delta H minus T, which is the temperature, times delta S, or the entropy. Organisms live at the expenses of free energy. During a spontaneous change, free energy increases, the stability of a system increases. At a maximum stability, the system is at equilibrium and can do no work. In 8.2, it also talks about exergonic or spontaneous chemical reactions. The products have less free energy than the reactants, negative delta G. Endergonic or non-spontaneous reactions require an input of energy, so positive delta G. The addition of starting materials and the removal of end products prevent metabolism from reaching equilibrium. 8.3. ATP powers cellular work by coupling exergonic reactions to endergonic reactions. ATP is the cell's energy shuttle. Hydrolysis of its terminal phosphate yields ADP and phosphate and releases free energy. Through energy coupling, the exergonic process of ATP hydrolysis drives the endergonic reactions by transfer of a phosphate group to specific reactant. This forms a phosphorylated intermediate that is more reactive. ATP hydrolysis, sometimes with protein phosphorylation, also causes changes in the shape and binding affinities of transport and motor proteins. Catabolic pathways drive regeneration of ATP from ADP plus phosphate.
The key concept for section 8.5 is regulation of enzyme activity helps control metabolism. Metabolic pathways must be tightly regulated by enzymes. Here's a visual of allosteric regulation. It has an inactive form, which is non-functional because the active site can't bind to the substrate. And here is the active form. And they alternate back and forth between the two. And if we want this enzyme to work, we want to bind an allosteric activator to it, which is going to enhance the stabilized active form so it works. And to turn it off, you must remove the regulator and bind the inhibitor to the inactive form for the same effect. A specific form of allosteric regulation is cooperativity. That is a substance bonding to a subunit enzyme that changes the shape of all subunits, which increases catalytic activity. So that is the substrate binding to the inactive form, and it's going to change all of them so they work properly. They each have an optimal temperature, which they work best, and an optimal pH, which they work best. This is a graph comparing the optimal temperatures for humans and heat-tolerant bacteria enzymes. And you can see that they have peaks, which they work the best, and then on either side they start to denature and not work properly.